Everybody see, please. Yes. Okay. I have to see my face.
No, the bigger.
Hey guys. Let's start.
Que se está reduciendo, como tú habías bien previsto, el tiempo de la discusión.
Okay, guys, let's keep up the 10-minute talk and 1-minute question. This is the second session, and I'm going to try to push you to be brief, because otherwise the people in the afternoon will have less and less time. So to be fair, let's use the time wisely. So I am the first speaker, so I'm going to be my own chair. And so don't worry, I think I'm going to get the talk soon. Yes. So actually, I'm very famous for giving talks of 2 hours, but because you have only 10 minutes, I'm going to give a super brief update on the recent result on our lab, which I hope you are familiar with, which is related to the hypothesis that the brain works near criticality. I'm going to show results from 3 recent papers, probably only from 2, and I have 10 slides. So I'm going to give you time at the end to ask for technical questions or things that are not clear here. But in case you miss that, the talks that the papers I'm referring to are listed here. This was one of my postdoc in Buenos Aires, in which we studied which kind of noise, which kind of fluctuations is brain noise, or the fluctuation that we see in the brain. We discovered an anomalous scaling at the resting brain activity, and you can read it in this paper in 2012. Second paper is another paper in Frontiers in Fractal Physiology by one of my master students, Enzo Tagliasucci, that now is in Frankfurt, and titles Criticality in Large-Scale Brain fMRI Dynamics, unveiled by a point process that we introduced in that paper in 2012. And the third paper is a paper a physical review paper, physical review letter paper of this year from another of my master students in physics from Buenos Aires, Ariel Jaimovici, that now is spending time in Trieste with Matteo Marsili. And the title is Brain Organization into Resting State Networks Emerge from the Colic Dome, this fancy word that you, I'm sure, hear of, at criticality. And so what is the problem? As I said, I have only 10 slides, I'm in the third one. This is the problem we want to study. You have a collective dynamics, which, as was discussed before, is the biological basis of everything you do. So everything you do has to be in the brain, as we heard before. And in particular, any behavior you have, I'm sorry I don't have the dancing lady here to accompany with the collective dynamic, but if you do neuroscience, you have to sort of convince yourself that everything that you see in this realm have to be embedded in these dynamics. That's a result that you have to accept in order to start discussing. But the question is that this ongoing dynamic, this is from myself, speeded up 7 times in a fMRI machine, in a magnetic resonance imaging machine, and whatever is red or yellow means that there is more activity there, neurons are more excited, and everything that is blue, neurons are less excited. So this, what I call brain meteorology, to give it a time and space scale name, have to explain these dynamics. And at the same time, recent technology allows you to have the connectome, the so-called connectome, which are the highways and the streets from where this activity is supposed to go. Not is supposed, must go. It cannot go outside these streets, okay? So the time to speculate and to make wild conjectures about how this problem is finished, because now you have the data and you have to solve what you have to add to this structure here in order to get this, and then make the connection between the three. This is the game we're playing for the last few years, and just to give you sort of a similar problem, which is less magical and less mysterious than the brain. It's exactly the same problem. You have the structure of the street, what will be called the streetoma, and at the same time you have a traffic engineer, and you have different phases. You have moments in which the streets are empty, almost empty, and moments you have a traffic jam. And you want to understand where the traffic jams are coming from. And also from the traffic, in the same way that before, you have that this activity is the short-term correlate of the dynamics, but also the dynamics can influence in long-term where the activity is happening in the street. Probably you have the same situation. The traffic engineer will make that this traffic jam will try to modify, adding more highways in where they need it, and so on, and they will cut streets that they don't use, and the whole thing is, in the long term, a similar problem. Although, in this case, you have to introduce, we know, cars with the dynamics of the driver. Drivers that have certain dynamics are more prone to introduce dynamical quenched disorder or temporal disorder, producing traffic jam that other kind of drivers and so on. This problem is very well studied in physics. There is many papers starting with K. Nagel's models in the 15 or 20 years ago. What I'm going to show you in the next remaining slide is to add here what I said that we don't know and try to see what we get. Okay? So this is then the problem we're going to study. We have the structure, which is the connectome. We are going to add some individual nonlinear dynamics to the elements. Okay? Just based on first principles, we know that in order to have different phases, these dynamics have to be nonlinear. Nothing will come out from linear dynamics here. So we can choose different nonlinear processes simulating neuronal dynamics. You can go and use very detailed models, but since you know that the emergence of pattern is not related to the detailed dynamics, because we believe in universality, you can choose a three-state simple dynamic from resting, excited, refractory, resting, etc. So it's a three-state serial automata from a neuron, and see what happens. So the idea is that depending on some control parameter, you will see some different phases. You have ordered dynamic in the brain covering the entire brain. Again, this is large-scale dynamics, so there will be like a cloudy day in my brain meteorology. This will be a sunny day, and very disordered, with few clouds in the sky. And this might be some place in between in which you have critical dynamics that we hope that will replicate the result that you see in the experiments with functional magnetic resonance imaging. Again, I emphasize that we are not doing an open experiment or an open simulation. We have experimental data that's contained these three papers I mentioned, but I don't have time to go into, in which we're going to compare what you see in the model and what you see in the experimental result. So the structure that we're going to use is the so-called human connectome at a resolution of about 998 nodes, meaning in different colors I paint here. These are three different projections of a brain from the side, from the top, and from the back. And you see each of these parcellations is one of these 998 nodes. The initial picture I showed to you with all this spaghetti covering the brain will connect each of these between them. So you have a coarse-grained representation of your brain in which you know that where this guy connects with. This guy is a parcel of about 1.5 cubic centimeter. It's a huge chunk. It's like having a map of London or a major city in which you have to have exactly all the streets. You have like a block of 5 by 5 or 5 by 5 streets, and that's a chunk that you put here, okay? And what is the information? The information is depicted in this panel A. In here you have the right hemisphere and the left hemisphere, 499 for the right, and each dot over there tells you which area is connected with which. So you have the adjacency matrix for each of these patches over here, which is given by the experimental result of the conic dome that tells you where these connections, real physical connections of the brain. And panel V tells you for each of these connections, again, this is the right, this is the right, this is left, this is the left. So these are connections between the left hemisphere to the right, and this is from the right to the left. This is inside the right, inside the left. For each of these, you have how much is the weight, mean how strong is the connection from one patch to the other. In traffic term, will be how wide is the highway that connects these two cities, or how much traffic we'll have if you put traffic on them. And so we have this structure, and we put some simple dynamic to connect each of these nodes. And as I said, as a matter of principle, if universality apply, almost any nonlinear rule connecting this node will have to should give you the same result, or approximately the same result. So any of these results I'm going to show should not depend if the brain have universal dynamic in the statistical mechanics point of view, should give you the same result independently of the nonlinear rule you use for the individuals. Okay, let's see what happens. The only parameter in the problem is this W. Okay? So the dynamics is the three-state dynamics. You have each connection with each connection, and you only have only W. And so this will be your control parameter. Your control parameter will be you update a single node, saying you are active or you're inactive, whatever you started the initial condition. And if your W allows to excite to whoever you are connected, the other guy will be excited, and so on and so forth. So the only parameter you have is a control parameter. If you move the control parameter, now let's define order. Order will be defined as the size of the largest cluster of activity, the size of the largest cloud you see in the sky. Today, order parameter in Amsterdam is 1. Okay? So if tomorrow I have two clouds, I choose the largest one, I express as a proportion of the entire sky, that's my order parameter of tomorrow, and so on and so forth. So I found that it's more convenient to choose the size of the second largest cluster, which define exactly the critical point. This is the moment in which the cloud is really as you move the threshold, the clouds start getting covering all your sky. So there is a critical point in this finite 998 nodes with all the caveats of explaining or not is this critical the rigorous point of view. And now if you choose this value, on the right you call it supercritical, on the left you call it, supercritical, subcritical. And now let's see the size in panel B, the size of your clusters, the distribution, the PDF of your cluster size. If you are at the critical point, is the red one, it's power law distribution of cluster sizes. If you are in the supercritical over here with a very high threshold, you have that all your clouds are falling exponentially in size, or all very small sizes of clouds in your head. And if you are on this side, most of the clouds cover the sky, so you have super clouds. The cloudy day over here is the blue one. And so you have indeed what you expect from this class of problems. And now let's compare with experimental. If you take a functional magnetic resonance imaging, the paper from Tagliasucchi, and you measure exactly the same in the brain of this individual during the dolce far niente, the resting state of the brain, meaning not implicitly asking anything or not explicitly asking response for the brain, you have that you have the black crosses. So you see that the black crosses, the experimental result, without adjusting any parameters, except to just going to the critical point, agree perfectly well with the model of the conic dome with nonlinear dynamic, trivial, very simple nonlinear dynamic, only when the model is at the critical point. conclusion of this comparison is there must be some relationship between my experimental result and my numerical result with the model, in the sense that I am only able to replicate this result at the critical point. You should read this paper. There are other things beside the cluster size distribution and blah, 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 that are found in this paper, but I don't have time to go into. At the same time, I would like to show you some other fundamental result, empirical, from Tagliasucci paper last year, in which there is indication that you have a phase transition in the resting brain, in the brain doing nothing, again, from experimental data, collecting data from functional magnetic resonance imaging at the brain. And basically, what he did was to plot the number of active sites in the brain, the order parameter in the same way that we defined it just a minute ago. The green points are the experimental point. The red one is the average of the green points, indicating that beyond certain activity, the system starts ordering, passing through a transition. And the green, the blue is the susceptibilities, meaning the standard deviation of the green points, which peaks approximately where the phase transition is expected. And most importantly, the resting time, the number of times that we observe the system, is higher near the critical point. And the bottom line of this, in my last slide, is that the resting state networks, which is, for those familiar with the literature, recently the hottest topic in neuroscience at this level. For those that are not familiar with this jargon, is basically the cluster activity that you see in the brain without asking the brain to do anything. This activity clustered in about 8 or 9 different patterns, and these are patterns that are related to, for instance, to somatosensory, this in particular, auditory, visual, blah, blah. Each kind of feature is represented by these blobs that you see here. I don't have time to go into detail, but these blobs are the signature of the class of activity that is correlated and is coherent in the brain. And as I said, it's like a fingerprint of a normal, healthy brain activity at rest. Now let's see what we need to do in order to match this activity. So this activity is from real experiments in functional magnetic resonance imaging, and these are 3 typical patterns that you find: a disordered, subcritical, an ordered, supercritical, and a critical. So if you compare this row with this row, you will see that indeed it agrees only when the system is critical. And if you read the paper, you will see more details of how strong is this agreement, only adjusting your control parameter to put it at the critical point. And I emphasize that there is no parameter in the model, and furthermore, if anybody have a bright graduate student with enough time, he can replace the 3-state model that is inspiring neuron and put a logistic map of any nonlinear map, and I bet you that you're going to get exactly the same result, meaning that what is important is that the system have the dynamical features of criticality and not the structure, because the structure is the same in the three results, and not the details of the dynamic, but the same universality. What is important is the dynamics at that level. So in summarizing what I said, I think that in the last few years we have been demonstrating that some fundamental large-scale brain dynamics is only replicated when you are near criticality. As in other critical phenomena, we should be interested in the future in looking at universality, which class of universality, which model we should be using. We should study more careful the anomalies introduced by the structure. I mean, the structure is not a lattice, it's a structure. For some reason, the brain has that structure. And we know that the quenching disorder introduced some phases, some rare phases, or some other people call it Griffith phases, which sort of pin the dynamics around particular points. That should be important, need to be studied. In biological issues, should be also interested in pathology, some we were mentioning before, coma, anesthesia, any other altered state of consciousness, and how these things are related. We particularly are studying in recent years a patient with psilocybin, with mushroom magic mushroom and LSD, using this as tools to investigate psychedelic state. And of course, there are philosophical issues that we should be paying attention. Thank you for your attention. Okay, and now there is no time for questions, so we should move to the next speaker. Which is Professor Nicole. I was kidding about the question, but I'm sure that we can talk later.
So my question is, when you don't have a criticality, what happens? A resting state issue, and you don't have resting states.
Actually, when you are not in resting state is defined more or less unsuccessfully as a state in which the brain don't have an explicit input, don't have an explicit output like
Yes,
now.
but if you
So-
are outside,
You are in resting state.
no, if you are not in resting state,
Somebody is putting you an input, and you should study as an engineer problem, which you kick it and you get a response. Exactly there. That's the so that's the engineering way. So you put a stimulus, you get a response, you analyze the linear response, non-linear, whatever. Okay? So, but that has been for the last 20 years. So the new thing is that indeed you can get information from a non-equilibrium system in this way. Okay? It's a bit what? We should discuss later.
Yes.
Okay? Yes, please.
Okay. Thank you. The title of my talk is synchronization in time discrete model of two electrically coupled spike bursting neurons. This figure shows examples of spike bursting oscillation of neurons. It's real neurons, and you can see this behavior. It is spike bursting behavior. It is clear that spike bursting oscillation have two distinct phases characterized by two different time scales. An active phase comparison a sequence of fast spikes, and a passive phase with slow variation of membrane potential. We have introduced a model for study this behavior. It is map, and here is discrete time, and here H is Heaviside function, and in fact we have two-dimensional map, non-linear map. Here you can see behavior in phase plane of these systems, chaotic attractor, which generate this chaotic spike bursting behavior. Now consider dynamic of this model in detail. Here, first of all, here X variable describes qualitatively the membrane potential dynamics of neuron. Y is responsible for total effect of all ionic currents. It's called recovery variable. The positive parameter epsilon is very small, and determines a time scale of recovery variable. Parameters beta, D, and J control the shape of signal generation. On the one hand, this model based on discrete version of Fiske-Nagumo model. Really, if this term is disappear, we have discrete version of very famous equation, Fiske-Nagumo equation. And another hand, for fixed y, when epsilon is equal 0, O very small, we have y is we consider as control parameter, and we have one-dimensional map, which demonstrate behavior similar for several regions of Y, similar Lorentz type map. Here they exist chaotic interval here. I forgot to tell about this function is cubic function and Heaviside function. In fact, when epsilon is not constant, is very small, we obtain fast slow two-dimensional map with discontinuity x when x is equal d. Now let us consider regimes which can demonstrate this model. You can see here another one regimes of this model. For example, spike behavior and phase play here. Substratial oscillation and close curve here, and periodic spike behavior, and in two-type periodic spike behavior, and chaotic regimes. You can see here different regimes which I showed before. This substratial oscillation and chaotic spike appear in this picture, and here another one. Now we consider two coupled of this model. Its systems mimics electrical coupled neurons by electrical synapse. Coupled by electrical synapse. C is coupling coefficient, and you can see this system is four-dimensional nonlinear map. We study this map theoretically using fast-slow dynamics approach. The main idea of this approach is following: when epsilon is equal 0, we have y1, y2 are constant, and we have two-dimensional system. It's called subsystem of fast motions. After that, and we study these systems. After that, we study so-called subsystem of slow motions. You can see here. After that, we put epsilon is not 0 and study dynamics. This dynamics like fast-slow dynamics. And first of all, let us consider dynamics of these systems. We consider y1 and y2 are control parameters. In fact, we have two coupled Lorentz type maps. Behavior of these maps depends of y control parameter here. This figure shows phase plane of these fast subsystems. There exist several type attractors on phase plane, chaotic attraction and stable fixed point. You can see it depends on values of y variables, which we consider as parameters now. You see three attractors, big attractors A and two small attractors, chaotic attractors and stable fixed point. For different y, we have only this situation. All this situation, all this one and this one. But in this figure, you can see phase portrait in where besides attractor, we put also unstable saddle type fixed point. You can see here stable saddle type node, unstable node, and saddle, and three chaotic attractors here. Now consider bifurcation. We change y variables as parameters and consider bifurcation. First of all, let us consider what is different between chaotic attractors A, this chaotic attractor, and small, big and small chaotic attractor. You can see different here. For small character is typically this type behavior is typically for chaotic attractors. You can see foliated structure typically for this attractor. For big attractor, you can see zoom, one zoom, another one zoom of the chaotic attractor, and you can see the structure. For big attractor, the situation is different. It's for this attractor typically densely covered by trajectory. You can see here. Zoom of this attractor. When we change y as parameters, for this attractors typical the following bifurcation. For fixed point, typically saddle node bifurcation, like this bifurcation when this fixed point and this point collapse and disappear, and all different situation when this saddle and this fixed point collapse and disappear, and for very interesting situation with chaotic attractor, how it disappear when we change parameter y. This atypical bifurcation for chaotic attractor, they destroyed through boundary creases that may be accompanied by appear gamma-clinic orbits to unstable fixed point and fractal basin boundaries. You can see this situation here. You can see in this figure. Boundary basin of attractor of fixed point has fractal property. After that, let us consider slow motions. What is slow motions? Because our system has 4 variables. And fast variables x in this case maybe belong to O stable fixed point, about stable fixed point, or belong chaotic attractor. And slow motions is different for these cases. You can see here typically we have 2-dimensional manifold for slow motions. When this trajectory cross this line or this line, we have in fast subsystem we have saddle node bifurcation and fixed point disappear. In this case, when you can see more complex behavior or slow variables, you can see when trajectory belong attractor, we have some this type behavior, some chaotic changing on variables y1 and y2. Now consider together this behavior. Now epsilon is not 0, and we consider dynamical of this in this case. Schematically, you can see this dynamical here and here. Let us consider behavior. Let us initial condition belongs to dimension slow manifolds. When this trajectory cross boundary, saddle node bifurcation of fixed points of the fast subsystem appear. After that, our trajectory going to fast motions of chaotic attractor of fast subsystems and slow varying of y. After that, contact bifurcation, boundary creases, and so on. After that, we come back to stable manifold. Result of this dynamics you can see here. You have this figure shows shows behavior of x1 and x2. You can see spike bursting chaotic behavior, two types. We study synchronization of this type of oscillation. We introduce value sigma, which characterize the ratio of the total time of coincidence of active phase of oscillation to the total duration of all active phase. You can see this formula here, and here you can see this it is sigma, and this coupling coefficient. It's for different epsilon. For different epsilon, when a coupling coefficient is small enough, we have very sensitive behavior of this value, and here we have synchronization. And also we study for understanding transition of the system between different states. We have applied technique of the theory of Markov topological chains. This we introduce several states of our system. X00 are both of neurons are passive. This X1, 0, the first neuron is active, and so on. And we calculate probability of the event that the first neuron state in passive state and the second active. You can see this probability in this figure in two cases. We define and we also calculate we calculate probability of the events that the first passive neuron becomes active and the second one is in any state up. And the same under the condition that one time step ago the second neuron was active, it's down. The influence of the active state of second neuron on the transition of one neuron from passive state to active one becomes significant beginning from some coupling strength C maximum. It's different, this line corresponding to difference between this probability. It is so-called, we call this...
Time is up.
Okay, we call this memory effect, and I almost finished. Some conclusion you can see here, and short conclusion, and I thank my courses, Maris Kurbash, he stay here, and my PhD student, Oleg Maslenikov. Thank you for your attention.
Very good. I think that there is no time for questions. Maybe at lunch we can discuss while we say thank you again. I remind the speaker that when I'm up, it's because your time is already up. So the next speaker is already. Is your talk already?
Yes, if you can help me.
It was in the
Somewhere.
Yeah, it was somewhere in anybody knows how to PDF full view?
Do that.
View?
Full screen.
Very good. implicit is this one, the red one, and this you go up.
Okay. Okay, today I want to talk about renewal fractionality in the brain, and this talk is in some parts related to the talk of Paolo Allegrini, because we work together in Pisa on this topic. And okay, a brief summary. I want to make a very short review of how it is possible to derive fractional diffusion equation from continuous random walk as a limiting, in the limit of long times. And I want to show the connection with the renewal processes, and in particular what we call fractal intermittency, and how we use these ideas for a data analysis method that we developed in Pisa, and using many results of found in the literature. And we use this method to say if a process is renewal, and if there is what we call a complexity index well defined. And then at the end, I'll give you some short ideas about fractional equation in physiology. And actually, I end with a question, in a sense that we can discuss later, perhaps. So, okay, let me recall what is continuous time of work. Continuous time of work is a model that is known from, at least from the 50s, and is a sort of mesoscopic model with trapping wells. So you can think about a lattice of atoms, in which there is a charged particle going from one well to the other. When the particle goes to a well, then it stays there for some random time, depending on the dynamics, and then it jumps over another well. And this is, let's say, we can use this scheme here of random walk, in which we have these random times, tau 1, tau 2, tau 3, that we call waiting times between two events. And essentially the random walk moves only when there is an event. We call this time intermittency. And essentially we can derive from here a master equation generalized in the standard Markovian one, because essentially we have here this n represents a sort of internal time. This is the Markovian version, and it is generalized by the fact that you have this integral here, depending on time, where the fundamental property is given by this psi, which is the waiting time probability distribution. And after some algebra, you get this standard form of the master equation, where you see there is especially this memory kernel here, related to this waiting time probability density function. Here, the bar represents the Laplace transform. And what is interesting is that when you have some scaling relation for the waiting time distribution and for the jump distribution, essentially you get in the runtime limit what is known as a time-space fractional diffusion equation. And here we call mu and alpha complexity indices, and we are particularly interested in this mu because it's something generated in memory in some sense. And okay, these are two particular cases.
Why you choose this time distribution?
Because it's something emerging from
time
here.
distribution is always
No, actually, okay, it depends. For us, it's only important that we have this in some scale of some range of scale, typically long range. So what happens in the short time scale is not really important. And okay, these are two particular cases: the time fractional diffusion equation, where the long range memory is containing this fractional derivative that is known like Caputo derivative. And here the space non-locality is represented by this operator here that is known as a Riesfelder operator. And this is a model for Levy flight, when you have a random walker that from time to time makes a long jump. Now, okay, my basic observation is that in all these, we can make this derivation if we have a renewal event. That means that the waiting times are independent, and typically is a condition that arises in many systems, nonlinear systems, like blinking quantum dots or liquid crystals, earthquakes, and for example, also in brain dynamics through the rapid transition processes of finger pulse. And essentially, the idea is that you have metastable states with a relatively long lifetime and short time transitions between states. And the interesting case is when you have this power law behavior, which we call fractal intermittency, and now we are referring to this as temporal complexity. Following, I think, ideas of Grigolini, I think is the first calling temporal complexity, this kind of. And anyway, okay, this is another example from turbulence. And okay, now I pass to what is the method that we used for the analysis of the EEG data for the human brain. We used, let's say, okay, there is a method to extract the events, the rapid transition processes or similar. And then what we do is to build 3 different random walks. The first one is called, we use what is called a symmetric jump rule, in which the walker moves only one direction. The second is the same but with symmetric jumps. And the third one is called symmetric velocity because there is a change in the velocity every time there is an event. And this is called also telegraphic signal sometimes. This is a sketch, these are sketches of the instantaneous velocity. To get the diffusion variable, then you make a sum over this instantaneous velocity. This means that, for example, here you have 1, then 2, 3, 4. Here you have, for example, you have a linear function, and then you go like that. And here you have a +1, 0, +1, 0, and so on. Okay, we studied the scaling of these 2 different scaling of these diffusion processes. And the first one is related to the self-similarity of statistical distribution. And we use a method called diffusion entropy for this. And the second is related is the scaling of the second moment. We use the well-known the trend fluctuation analysis. Note that for fractional motion delta, that is the self-similarity of the statistical distribution, is equal to the second moment scaling, but is not true. This is not true in general for diffusion driven by renewal events. Here is a sketch of the theoretical results when you have renewal events. Now, what was the idea? The idea is that we have a sequence of events from real data. We make a simulation taking essentially the building the diffusion variables for the 3 random walk, and then we compute the scaling, delta and h. Now, if you get results that are in agreement with this picture, these 2 pictures here, then we can say this is a sort of reasonable, not a theorem, but reasonable. We can say that we have renewal events, and also we have a robust estimation of the mu, of the parameter mu, that to us is a parameter that reveals in some sense the complexity of the system. For example, when you get this h equal and delta 0.5, that is related to mu greater than 3, typically, here in this condition, essentially is for us is a condition in which the system has a low complexity. That's the idea. And yeah, okay, this is the same, more or less the same picture of Allegrini, so it can be very. This is a particular application in which we are able, using this method, to distinguish between 2 different states of the brain. That is the conscious state, that is complex, with the H075, and the unconscious state given by the deep sleep condition, where we have normal diffusion. So this means that here the brain is integrated and here is, in some sense, segregated, but okay, we can discuss on more detail this if you are interested. As Allegrini told before, this difference is not evident from the avalanche size distribution. Okay, now, okay, I have time?
10 minutes already.
Okay, 12 minutes then.
We
Right.
have 3 for
Ah,
you.
3, okay, 3, yes.
2 for us.
And okay, I want to explain what are the points in favor of this kind of approach. Okay, the first one is that why don't we estimate the mu from the waiting time distribution? We use the diffusion scaling. Essentially because when you have noise, the waiting time distribution can give a wrong result. Like, for example, we made a model in which you study superposition of Poisson and non-Poisson events with power law distribution. And what we get is, in this example, what we get is that if you use a fractal intermittent model with mu equal to 2.05 and mixed with Poisson events, then there is the merging of an apparent scaling given by 1.6. That, however, is wrong. But we are able to find this mu equal to 0.05 using the diffusion scaling. Another point is that, for example, using the complexity index mu on a series of a sequence of events is something that is more reliable with respect to classical measure like the fractal dimensions. When you have a point on the line, you can measure the fractal dimension of this point. What happens is that if mu is less than 1, then you have this relation here, because the fractal dimension cannot be larger than 1. This is a theorem. But if we have mu greater than 2, then the f is always equal to 1, and you don't see any difference between mu equal to 0.5 or mu equal to 0.8. But if you use our analysis, you can distinguish these two different cases. And okay, now I am at the end, near the end. Just two examples from physiology in which there is the use of fractional equation. This is a model from Anastasio in 1994 in which he used fractional calculus, use some kind of circuit model with always with the linear elements, but with fractional powers of the Laplace variable, not integer powers. And essentially, this is why he did that, in order to give an interpretation of the data. And the same is happening recently. That was a paper in which it is the group of Magin, is the name Magin, right? That you suggested me, I went there and I discovered that they tried to use fractional calculus to interpret the data from brain image processing. And here, this is my limitization. There is a diffusion term, and they use this generalization of the Bloch-Torre equation with the fractional derivative in time and in space. And because essentially this is thought to be related to the strong immunogenicities in the water diffusion in the cell and in the environment around the cell. Okay, this is my conclusion. Essentially, this talk was just to give some spots in order to say, okay, we have the continuous standard work that is the basic to us. It's possible to derive fractional diffusion equation in the limiting or in the limit of long times if you have a temporal complexity. And then we use continuous time works as a method of data analysis in which we discover that there is a temporal complexity in the brain. There are fractional equations in some physiological system related always to neuron dynamics. So I end with a question: is it possible to derive at some level a fractional model for the brain? And that's all. And thank you for your attention.
There is a, thank you. There is 30 seconds for a question while they're putting the next speaker's machine. There is any question?
Is there room for a special issue on fractal
Yes.
calculus? Question.
The question, what is the answer?
Yes.
We have, yes, yes. And,
Well, our chairman can give the question and the answer. Thank you very much again. The next speaker is Dr. De Archangelis from Napoli. Excuse me?
Professor.
Professor Dr. Lucina De Arcangeli from the University of Napoli. She is always invited to contradict that widespread rumor that there is not beautiful women in physics. So that it was in response to your professor. Profesora, usted tiene, you have 15 minutes.
I know,
You
I know,
can
I
use
know.
You can use the 15
By now
minutes.
I know very well.
as-
Don't
as
worry,
you
relax,
wish.
relax.
You can use 10 minutes for your talk and 5 minutes for questions.
Okay,
15 minutes
okay.
for talk and nothing question. Thank you very much.
Thank you. So I will present to you today a very short summary of some results that we obtained in modeling from a statistical physicist, because I'm a statistical physicist in critical phenomena, and the attention of this talk would be mainly on learning, so would be on the kind of activity the neuronal system has when not in the spontaneous state, when there is a particular simulation, as the question that was asked before. So by now, I think it's known to most of you that in 2003, spontaneous activity has received a lot of attention in the literature because Dietmar plans showed that spontaneous activity is made of bursts of activity at electrodes in a microelectrode array, and that these bursts, analyzed at a finer temporal scale, give rise to, are made of, of avalanches whose size and duration are distributed as a power law. The appearance of a power law is something that has attracted the attention of physicists in critical phenomena, because the absence of a characteristic scale, both at the spatial and the temporal level, is a signature of the system acting close to a critical state. So this is why people studied a lot this system experimentally, in vitro, in vivo, in the rat, in the monkey, in dissociated neurons, but also in modeling. So the idea was to try to reproduce with a simple physical model. So this is a model that belongs to a different class with respect to usual model for neuronal systems where you have a partial differential equation with time derivatives and so on. This is a model inspired in self-organized criticality. That means the model is made of neurons, sites, links that are synapses, and once you stimulate a neuron and the neuron gets to a particular membrane potential value, distributes charge, so distributes its potential to the connected neurons proportionally to the strength of the synapses. The synapses are directed connections, so the matrix is asymmetrical, and the first equation tells you how this potential evolves in times. Once a neuron fires, goes back to a resting potential and gets into a refractory period, and this activity can propagate involving a number of neurons, very variables. But then when it stops, you can stimulate again to have another event. So you can actually generate with this model an avalanche activity. And the other feature of this model is that connections are not static in time, but they evolve. We apply Hebbian plasticity. So when a connection is used, it gets strengthened, otherwise it gets weakened, and it can even disappear. There is the phenomenon of pruning. So the only thing I want to summarize is that via this model, but then as also the other models like Levin et al. And Millman et al., you can actually reproduce the scaling feature of avalanche activity. And the nice feature of our model is that you can actually tune different features, and if you believe in critical phenomena, you should expect that the typical, the features of the network, the neuronal behavior, and the type of initial conditions, and the distribution of strength, all these features are irrelevant with respect to the scaling behavior. And this is indeed what we find. We find that the scaling exponents do not depend on the kind of network, of the strength of the connection. It's truly a critical system. But then, with this model, it's nice that you can also analyze the temporal organization of avalanches, and in also previous talks, attention was given to the waiting time distribution. That means the distribution of quiet times between the end of an avalanche and the beginning of the next one. And this distribution is interesting in stochastic phenomena because if it is an exponential, really you have a Poissonian process. But as soon as you have a Pavlov behavior, there is a nice paper by Utsu et al. That is not very well known because it's in the seismology literature, that shows that as soon as you have correlations in the rate, you can actually prove analytically that there is a power law behavior in the waiting time distribution. And these are the experimental data by plants on the culture of rat cortex, and you see there is an initial power law regime with a non-monotonic complex behavior followed by a bump, and this complex experimental behavior can be reproduced by the model by appropriately inserting some feature of the neurons. But I don't want to get into these details. I want just to tell you that the temporal organization of avalanches gives indication of a dynamic balance that the system is able to establish between excitation and inhibition in the system. By dynamic balance, I mean that I'm not talking about the quench excitation and the quench inhibition, that means the percentage of synapses, those are quenched, they don't vary in time. It's the system itself that goes through phases where you have a lot of activity, avalanches are correlated and close in time, and then the recovery periods called down states where the system erases memory of past activity and goes into, a quiet time that is more or less long. And I here want to mention that this behavior found experimentally in cortex slice was also confirmed by a recent paper of Chialvo and co-workers that was published into a paper of the special issue in honor of Gerhard Werner, edited by Paolo Grigolini, where this idea of balance between excitation and inhibition is also found at the scale of the entire brain. So the brain is a system that somehow is able to balance itself, or establish this homeostatic mechanism of regulation between excitation and inhibition. But these are, the analysis of the avalanche activity, that is the activity measured into the spontaneous state. Okay, so our question was, what happens if I have a model that can reproduce all these behavior, these features of spontaneous activity? What happens from the learning point of view? Is this network able to learn, just in the hope of understanding the complex behavior of a real brain that actually learns tasks? And in the process of this research, we realized that what is crucial in the learning procedure, in the learning mechanism, is the role of inhibitory synapses. And here I summarize two experimental data that turn out to be crucial to have a good efficiency in learning. One is the observation that in real brains, mammal brains, you have a percentage of about 20–30% of inhibitory synapses. Why? Question mark. Experimentally, the science paper by Bonifazi et al. Have combined fMRI measurement with optical measurement and have realized that inhibitory neurons are hubs, so are highly connected. They don't have a low connectivity, but they all have a very high connectivity. And then finally, there is a Nature by Royer, who actually shows that Hebbian plasticity is a very simple paradigm. There is a more complex homeostatic plasticity, because if we induce strengthening into excitatory synapses, let's say, nearby inhibitory synapses are weakened, and vice versa. That means locally, the system tries to keep constant the average strength of connection. I call this homeostatic plasticity. So let's now come to the learning procedure in our model. I have my network. This is a scale-free network as measured by Egui-Lutz, the group of Dante Chialvo, and I choose two neurons, the red one, that are my input neurons. In these neurons, I put these neurons at the firing threshold, and this is the state 1. Otherwise, they don't fire, and this is the state 0. So I translate the neuronal behavior into a Boolean variable, 0, 1. And I want my network to learn Boolean rules and or XOR or a random rule with a number of inputs. So the two red neurons are connected via the same number of connections to a single black neuron. I have a pointer, a single black neuron here, that is the output neuron. That is the neuron where I monitor the output, the response of the system. And then I can actually apply several rules, and I see what is the output, the answer that the system gives me. If the answer is right, I do nothing. If the answer is wrong, I change the strength of the synapses. I apply plasticity in a way that was first proposed by Bach and Chialvo, but then we slightly modify this because we introduce a spatial dependence in the strength of the modification. And what turns out is that if the system has to learn a single rule, so here is the XOR rule that for people in the field is usually a very difficult rule to learn, you see that the system after a certain time is able to give right answer and then arrives to a stationary level that is the level of performance into to the task that you have applied. The different colors correspond to different strengths of the plastic adaptation. And so what is interesting is that the stronger the plastic adaptation, the sooner the system starts to learn, but then the final performance is lower. And that means is that if I make very slow quasi-static adaptations, the system takes longer to start to give the right answer, but then finally it gives a better performance, which is in agreement with the experimental paper of Lewis et al. That came out of class, where by fMRI they measured the plastic adaptation, the modification of the network in patients doing a task, so real humans, and they saw that those people who had very slow modifications of the functional network were the people that at the end were giving the best response, the best answer to the learning. But then, so we tuned all the parameters better, the larger the system is easier to learn, but then I want now to go into multitasking because this is what is interesting. So what happens if I want to learn more rule in parallels? After all brains try to learn more than one rule at a time. And then this is what happens. And the first interesting result is that, as you see in the previous graph for a single rule, these curves are basically monotonic. That means you always improve in time the percentage of your performance. But then, if you instead instead you learn more rules at a time, here I have 2 rules, you see that the system starts to learn first the easiest rule, the OR in our case, but then as it tries to learn also the second rule, here the 2 lines, the blue one and the red one are superposed, it automatically starts to forget the rule previously learned. So to learn more things at the same time, learning goes through forgetting. It's just an interplay in time. So you think you have learned something, but then as you face a new challenge, you partially don't understand anymore what was clear to you before. And this is found, this behavior, for all rules. I mean, there are very wiggy patterns that are not statistical fluctuations. These are really learning and forgetting of the system, and this is verified also for 3 rules. But then we wonder, what is the role of inhibitory synapses? And so you see the curves for the final performance of a system that has to learn 2 rules, AND and XOR. And as you see from this graph, is that you start from a 0% of inhibitory synapses. As soon as I increase inhibitory synapses, the performance improves. So here in the graph on the right, you just see the asymptotic behavior of the system. What is interesting is that this performance has an optimal value for 20-30% of inhibitory synapses, but as soon as you have a larger percentage of inhibitory synapses, the performance goes down. So it seems like this 20-30% is really an optimal percentage. What does correspond to? Physically, we analyze the system that learns, and we see that in this system the shortest path is shorter and the backbone is largest, but then, this is my last slide, this is the most interesting, probably, interpretation of the phenomenon. This is the avalanche distribution for a system that learns. So not the spontaneous activity that has a power law behavior, but just the avalanche that gives the right answer. So you see that for 0% of inhibitory synapses or 10%, the avalanches involve the entire system. To learn, all the neurons in the system are involved in the activity. As soon as I increase the number of inhibitory synapses, you see that you can learn actually involving fewer and fewer neurons. So you don't need to have all neurons involved. So the ab-inhibitory neurons confine activity, but you still have a good performance, even better. So how can I characterize this? So I can interpret this distribution of avalanche size as the probability to have a configuration of the system where S neurons are firing and N minus S are not firing. So I can define an entropy associated to the system with the famous Shannon entropy formula, and I can characterize by this entropy the variability in the response of the system. By increasing the number of inhibitory synapses, the variability increases. At the same time, the excitability of the system, the capacity of the system to propagate activity, can be measured by the average strength of the synapses. And of course, since inhibitory synapses are negative, the average strength decreases by increasing the percentage of inhibitory synapses. So the final outcome is that if I define a functional that is the product of variability and excitability, you see that this functional is maximized precisely for the percentage of inhibitory neurons that is observed in mammal brains. So our network, brain network, is organized so that it's made of such a percentage of inhibitory neurons that maximize at the same time makes a compromise between variability and excitability in the response. So I will just leave the conclusions and
It's okay. It's okay. Don't do it. Don't do it. So
I'm done.
I'm now doing my inhibitory function, which is to
Yeah,
say
but I
thank you.
self-regulated.
Self-organized is good, but well-organized is even better. So there is no time for questions unless you want to use the time while you set your computer for a quick question. Anybody? No? Okay. We thank you again. And now it's Professor Chavez will have his 15 minutes. There you go.
Okay. Thanks. I would like to first thank the organizers for inviting me to this meeting. At the beginning, I was planning to talk to you about brain connectivity networks, but then Marisa told me, no, Mario, please. The objective of the meeting is try to know each other and try to know what you do more precisely. So I will start firstly by presenting me. The place where I work in the Brain and Spine Institute in Paris, located at the largest hospital in Europe. It's the Pitié-Salpêtrière Hospital. And we are more or less 600 people, researchers, postdocs, PhD students, technicians, engineers, monkeys, rats, and zebrafishes. The institute is this one. It's a multidisciplinary institute. Mainly, you can find groups working on genetics, molecular biology, a lot of molecular biology, in vivo and in vitro electrophysiology, optogenetics, and we are a very, very small group, about 50 people, working on signal and image processing, and we apply all our stuff to different diseases. The institute, different floors are working mainly on Alzheimer and Parkinson, sclerosis, epilepsy, spinal cord injuries, coma, many problems related to consciousness as well as sleep. So the main framework of my research is something related to what Professor Chialvo already explained to you before. I try to understand how the anatomical network of this fancy word connectome, for me, is a kind of physical scaffold of the brain where the dynamics take place. So try to understand how these two words are related in order to produce what people call brain network. So some people try to represent the connectomics of the functional dynamics as a matrix of interactions. And we are trying to relate this matrix interactions to different diseases, try to understand how it works, why it works in a certain way, and especially in case of diseases. So the observed phenomenon that I'm interested here is the fact that synchronization of dynamics, the synchronization of dynamics of some areas that are not directly connected. Now it's like a cartoon. Different brain areas are more or less specialized. And if everything is okay in your brain, in a healthy brain, the whole system acts a kind of big orchestrator, and the symphony is more or less well. But you can imagine that there are some areas that are far away on space, and nevertheless they are synchronized. They seem to play together. And what is interesting that is in some cases, these areas are not related by a central coordinator. There are some hypotheses about the role of a central coordinator, especially for the deep structures, some internal and very old structures from an evolutionary point of view, thalamic structure, nucleus, and deep nucleus, who may play a role of central coordinator. Or other hypotheses talk about the thalamic and hippocampus structures and their roles of internal relays. So there are some works, very nice works, from this group and also from the group of Palma de Mallorca, who proposed the following. They observed that in a very sample model, they coined the word the zero lag synchronization between neural systems. And what is that? So they observed that in a very sample configuration like this, they observed a synchronization between neuron 1 and neuron 3, but the dynamics of both neurons are not synchronized with the dynamics of neuron 2, like this. Each bar represents a spike discharge. So they associated this phenomena to a thalamic, the role of the thalamic delay in the brain. What is interesting in this stuff is that the zero lag is not the effect of external forcing. For instance, if you consider these two systems, of course they interact, but they are not synchronized. If you put an external, let's say an external driver, an external forcing, you can see that with this model, they are not synchronized. What is important is the transmission relay, the role of the transmission relay played by this deep structure, thalamus or hippocampus. And in this case, you know that the information can flow, and it's only under this condition that both areas, these areas, can synchronize. Again, this guy, Golo from Palma de Mallorca, and I recently learned that now he's in Australia. So we decided to play with this, but using a very sample model. I think that all of you know or have heard about the Kuramoto model. It's a very current model used to study synchrony in different things, from fireflies to menstruation cycles in females. So it's a very sample. The general Kuramoto model is like this. You have an internal frequency, an eigenfrequency. You have a coupling parameter. And here, this A codes the connectivity matrix between all the elements. And this is the phases of the different elements. But what is interesting in this complete model, the general model, is the inclusion of this parameter called frustration parameter. What is the role of this frustration parameter? You can see, I mean, it's very intuitive. For alpha different to 0, the oscillators cannot obtain, I mean, you cannot observe a full synchronization. The exact synchronization, the exact phase synchronization is avoided for two oscillators that are connected. But there are more interesting properties for this model. The fact that when your parameter alpha is comprised within this range, you can observe some phase clusters. And if this parameter is larger than a critical value, synchronized states are unstable. Some people call these unstable states chimera states, but for me it's not very clear what they are, and my humble opinion, are not very well defined, so I won't get into this detail of the unstable states where the synchronous and incoherent states coexist. They are synchronous for a period. Probably. For me, I mean, I have not fully understood these states, because they are synchronous for a while, unsynchronous for a while, and then synchronous again. And the distribution of times, I think that is a very challenging field of research. But so I will concentrate only in this case. So we were playing with toy models at the beginning of this study. We used this small graph, and we put this graph on the Kuramoto model with frustration parameter. And as you can observe, this is the phase, the sinus of the phase for the different oscillators. And you can observe how the phase evolves according to the frustration. Nevertheless, for instance, if you consider this value, you can clearly observe clusters. I mean, these two nodes are synchronized, these two nodes, which are not physically directly connected, synchronized. But here, for this value, if you make a zoom, you will observe something like this. I mean, the clusters are very well distinguished. It's only a question of scale. So that was our first observation. From a more theoretical point of view, I mean, you can, for very small values of alpha, this parameter, we can linearize the system and obtain something like this, where L is the graph Laplacian of the graph, and Ki is the number of connections for different nodes. So for the sake of simplicity in the analysis, we consider these conditions, and we can obtain something like this. We can solve the system like this, where this K is the average number of connection. This is the matrix representation of the whole system. And if you, you are curious enough, are patient enough, you can get the exact solutions for some toy models. For instance, here we proved theoretically that the phase of these nodes are identical. So, but if I ask you, what can you observe from this graph? It's very intuitive. You immediately can say, oh, the graph is symmetric. There is some symmetry on this graph. And so also we tried, for instance, from another type of graph, the bed lattice. And again, when they are coded, the nodes share the same color if they are synchronized. So we were happy to see, it was an observation, that the symmetry seemed to play a role on the synchrony. And it's the same for a chain. The nodes on the extremities synchronize and synchronize each other, and again, and again, and again, and the only node that synchronizes is the center of the chain. So that was an observation, and you can solve the exact system for this kind of models. But for more complicated graphs, we needed 1.5 years of computation, discussion, and so on. And so very, very fast, I won't get into the whole details. We consider the notion of automorphism. The automorphism of a graph is simply a permutation of a vertex such that the properties of the graph is preserved. So you can reflect, and the graph is preserved. For instance, here is an example of some permutation of the graph. If you permute this series of nodes and then you transform it like this, you will conserve exactly the same properties of the graph. But what is interesting with this permutation matrix is that if the permutation matrix is applied to the Laplacian, you get again the Laplacian itself. And that was the key for solving our problem. Our problem, after some algebra, we could obtain this singular system where here you have the permutation, any permutation operator, the Laplacian and the phase expressed as a vector. And if you leave free one variable, you can consider a new system, and we were happy to obtain the following. That all the phases of symmetric nodes, the phases of symmetric nodes, what I mean by symmetric nodes, the nodes that are related by an automorphism are identical at any time. So if they are identical, there must be some synchrony. So fortunately, we have the chance in my group to have access to different kinds of data. So we took data, anatomical connectivity, the connectome, and we used this graph as the connectivity matrix in the model, and then we put it in the Kuramoto model, we run the model, and we measure the difference of the phases of each node. And of course, we compare it with functional data, again the data from the fMRI obtained during the resting state As Professor Chiaro explained, the resting state is relatively simple to obtain. Subjects are asked to relax, to think about nothing. For 10 minutes, it's not easy, because if I ask you to think nothing for 10 minutes, you begin to wonder.
Can do that.
Sorry? Yes, it's not easy. It's a very challenging task to think about nothing, because immediately you begin to wonder, to imagine, and so on. And so, this is the results. Here you can observe in a logarithmic scale the difference of the phases, of all the phases in the graph. And here you can observe the highest coherence obtained for the graph. So what is the result? That symmetrical nodes with similar phases obtained through the model, I mean nodes in this region, expressed a very, very large coherence from the functional time series. And let me finish by a simple example of what we observed. For instance, here we consider two regions of the brain, the red and blue. They are spatially closed, and they were related by a physical connection between them. Nevertheless, you can observe that the signals, the time signals obtained from the fMRI are not synchronized. But if we measure the fMRI activity in the area coded by green, which is far away, especially far away from this other region, and is not related, directly connected with the blue one, you can observe the high synchrony observed between these two regions. So, as a conclusion, we put the hypothesis that the symmetry can play a role in the brain dynamics, and of course, from a more practical point of view, our method can be used to detecting symmetry in the nodes. But I must say that the method cannot provide information about the total number of automorphism. Here is simply another example of a graph, and how the nodes coded with the same color are not with the same phases, which correspond to, I mean, this graph is used as a benchmark in automorphism theory. So this work was made with some colleagues from my group, Fabrizio De Vico and Jacques Martinelli, some friends and colleagues from Queen Mary University in London, Vincenzo Nicosia, who is the main author of the paper. If you are interested in the activities of our work, you can visit, please, our site. Sometimes we put data, papers, positions, etc. And probably you will be curious to know what I put lasagna here, because lasagna is the financial support that we receive from the European Union. And here are some papers where we have explored the brain dynamics with this framework of complex networks. This is the thesis review letter about the symmetry and synchronization. But we have some other papers about modularity in epilepsy, and here the modularity in fMRI. Thanks for your attention. If you are interested in other activities related to the European project, please visit our site or follow our Twitter account. I'm in charge of the Twitter account, so we can exchange something. Thank you.
Thank you again. And as you see, we are running behind the program just half an hour. So now it was supposed to be at 1 o'clock, the lunch. But because we are delayed, we are continuing with the next session, which Matthias is the chairman, right? Okay, that was a joke. So, what is the plan?
Yeah, so the lunch is served in the room just next to it. So we have to go out and go around and learn.
And at what time we reconvene?